Hello everyone, welcome back to Dr. K PREM-PRIMER YouTube channel. In today's lecture, we will talk about construction of M13 cloning vectors. Construction of M13 cloning vectors. There are 5 types of M13 cloning vectors. They are nothing but M13mp1, M13mp2, M13mp7, M13mp8, and mp9. In today's lecture, we will cover the M13mp1. So M13mp1 is nothing but addition of genetic selection marker, that's a N-terminal region of beta-galactosidase, that's alpha peptide and its regulatory element addition to the M13 genome. Here we are adding alpha peptide region and its regulatory regions to the M13 genome. That is nothing but M13mp1. You can see. You see, this is a M13 genome. M13 genome, this is a replicative form, which is in a double-stranded, and addition of alpha peptide, that's a lag Z prime region, along with the promoter and operator. When you add those regulatory elements and alpha peptide region, encoding region, that's a lag Z prime region, then that M13 becomes M13mp1. So in this lecture, we will see how the lag Z prime region is added to the M13 genome, right? And before going into the lecture, I want to tell you that unless you understand the M13 genome organization and their features, it is not easy to manipulate the genome. Hence, we will briefly discuss about the M13 genomes and its features, then followed by the M13mp1 vector construction. As you all aware that M13 is a male-specific bacteriophage, means it will infect the gram-negative bacteria, which is having the phyli. That's a phyli, which is involved in the conjugation of plasmids or conjugative plasmids. And M13 have single strand closed DNA, single strand DNA, right? And its genome size is, you see, M13 genome is single strand covalently closed, and it is a positive type. And the size of the genome is 6,407 bases or nucleotides, since it is a single strand. And if you see the genome organization, there are 3 functional units. The first one is called as FTR. FTR means frequently transcribed region. Frequently transcribed region where protein 2, protein 5 are required for the, and protein 10 are required for the replication of the M13 genome. And the 7 and 9 are required, they are the minor capsid proteins. And the 8 protein 8 is the major capsid protein. So these are frequently transcribed region. They are frequently transcribed genes. Hence, that region is called as a frequently transcribed region. There is a second region. There is a second unit that's called as less transcribed region, LTR. Under LTR, there are 4 genes. One is gene 3, gene 6, genes 1, and gene 4. So gene 3 encodes the absorption protein, and gene 6 encodes the minor capsid protein, And gene 1 and gene 4 are required for the morphogenesis and assembly of the phase. And third functional region is called as intergenic region. Intergenic region. The intergenic region does not encode or have any protein encoding gene. Instead, it has two signals. One is origin of replication. You see, this is origin of replication required for the replication of M13 phase, positive or negative strands. And there is a packing signal. That signal required for the assembly of the phase. So this is all about the genome organization, right? Then you see every gene is very much tightly packed from gene 2 to gene say 4, they are tightly packed, right? So what are the attractive features of M13 genome? Why we want to consider this as a M13 cloning vector? Because it is a convenient source of single strand DNA. As I told you that single strand DNA is very, very required for the genome or DNA sequencing by dideoxy method. So chain termination method requires the single strand DNA. So single strand DNA of the insert or whatever you are gene cloning, if you get, then that will be easy for to know the structure of a gene or sequence of a gene. So by using the M13, we can produce the single strand DNA of DNAs. And also, more importantly, it can produce 5 to 10 microgram of single strand phase DNA for 1 ml of bacterial culture. When you transfect the bacteria with this M13, so with 1 ml of culture, you can get the 10 microgram of the single strand DNA. And it is easy to isolate also. And unlike lambda phase, it doesn't have the packing constraints. You all know lambda phase can only pack between 37 to 51 kb whenever genome size is that. If it is above that or below that, it cannot pack. So, but here no packing constraints. Third one is M13 does not kill the host. So these are the attractive features of the M13 to consider and to develop as a cloning vector. Fine. Where you want, as I told you that the genes from 2 to 4, they are very tightly packed. And you cannot use any gene of the FTR or LTR for gene manipulation, right? So they are very, very essential for the phase life cycle. So we need to search for a region which is not essential and also allows the manipulation of genome. That's nothing but intergenic region. Cloning site in M13 genome is intergenic region. You can see this is the intergenic region whose size is finite base pairs having the have a origin of replication and packing signal. So without disturbing the packing signal or origin of replication, if you can manipulate here and if you can establish a linkers and genetic selection markers there, then this can be used as a cloning vector. When you want to convert the M13 genome into a cloning vector, it should have a following features. You see, when you want to convert M13 to be a cloning vector, then it should have origin of replication, autonomous origin of replication. Yes, it has origin of replication. Second thing is that polylinker or MCCS means restriction sites. If you can develop a restriction site in the intergenic region, unique restriction sites, then with the help of unique restriction sites, they are called as multiple cloning site or polylinker. with the help of them, you can clone your gene of interest into the M13 vector. Then third one is genetic selection marker. Unless you have a genetic selection marker, unless you insert the genetic selection marker into the M13 genome, you cannot distinguish between the recombinants and non-recombinants. Hence, we have to include the genetic selection marker also. And also, we have to remove the unnecessary restriction sites present in the genome through the in vitro mutagenesis. When you remove the unnecessary restriction sites, then only unique restriction sites will be there in the polylinker or MCC region that allows the cloning of your DNA of interest. So when you want to convert the M13 genome into the cloning vector, you need to have a independent origin of replication. It is there. And we have to include the polylinker or MCCS genetic selection marker to screen the recombinants from the non-recombinants and the removal of unnecessary restriction sites from the genome. Then what are the applications of the M13 vector? You see, applications of the M13 vector are isolation of single-stranded insert DNA for sequencing. As I told you in the introduction that single-strand DNA is necessary for the sequencing if you want to use the dideoxy method of DNA sequencing. So when you clone your gene of interest into the M13 vectors, you can get this single-strand DNA that can be used for the sequence. Second application is in vitro mutagenesis. When you want to change the certain bases in the genome, that also requires the single-strand DNA. And the probe preparation. Probe is very, very essential to detect the macromolecule RNA or DNA through the southern blot or northern blot. For that probe preparation also, you require the single-strand DNA. So when you clone your gene of interest into the M13, you can sequence it and you can change it, or you can use that as a probe. So these are the advantages you have when you clone your gene of interest into the M13 vector. So here you can see one by one in M13-1, they added the genetic selection marker, and M13mp2 they added the polylinker, and also they removed the polylinker, only one ecoarm site. Then M13mp7 they added the many sites for cloning and also removed the unnecessary restriction site. Now we are going to see construction of M13mp1. See, to construct the M13mp1 vector, we have to take the replicative form of the M13 genome. Replicative form means double-stranded DNA. When a M13 enters into the E. coli, M13 genome enters into E. coli, immediately single-strand DNA converted to the double-strand. That is called as a replicative form of the DNA. You see, this is a replicative form of M13. So you see, as I told you that intergenic region is a region allows the DNA manipulation. So when they sequenced it, they found that there are 10 BSU1 restriction sites across the genome. So by using that one, by using those sites, they did the partial digestion with the BSU1. Partial digestion means digestion of few or one site with the restriction enzyme so that you get the linearized M13RF or replicative form. You see, rest intergenic region have two BSU1 site. And when you digest the M13 replicative form with the BSU1, and if you get a linearized molecule and you do partial digestion to get the linearized M13RF molecule with the BSU1. See, BSU1 is digesting only one site in the intergenic region and it forms the linear M13RF. M13RF. It's a linearized M13 RF. So linearized M13 replicated form is ligated with the N-terminal region of beta galactosidase, which is having the promoter, operator, and the lag Z prime region. Lag Z prime region. So here, a DNA fragment which is having the regulatory elements that are nothing but promoter and operator and the lag Z prime region. So, which is digested with the N3, both linearized M13 and the lag Z encoding nucleotide region are ligated, blunt-end ligation here. Blunt-end ligation. So once the blunt ligation happens, then that mixture, the linearized M13 and alpha peptide encoding nucleotide region, the transformation mixture is transformed into the E. coli, which is having the omega peptide region in the genome. So then transformed E. coli are plated on a media which consists of IPTG and XGOR. Right? So the plaques are if you can see a blue flakes on the plate, that indicates that reconstitution of beta galactosidase. How reconstitution has happened? Because lag Z prime region from the vector and omega peptide region from the E. coli genome, they club, and then in the presence of IPTZ and XGOL, the plaques looks like a blue color. So you can see blue plaques, that's an intragenic complementation, produce the active beta galactosidase, right? Alpha peptide, which is ligated with the M13 RF, right? Then it forms a kind of a genome. It is a ligated one, and that is transformed into the E. coli, right? And that E. coli is having the omega region of the beta galactosidase. So when there is a active, two components, one component alpha peptide from the M13 genome and omega peptide from the E. coli genome, when they are produced under the IPTG, they are reconstituted to form a functional beta galactosidase. When functional beta galactosidase is there, and under IPTG and XGOL condition, the plaques looks blue color, right? It means that the M13 is having the alpha peptide region encoding DNA into the intergenic region. So M13mp1 is having alpha peptide encoding region in the intergenic region. Then only blue plaques are formed. Whereas if you see white plaques, it indicates that self-ligated linearized M13 RF. So if this M13 RF is self-ligated, then you get a blue white plaques. But if it is if the M13 RF is ligated with the lag Z region, then it forms the blue plaques. So this is what is called as the M13mp1. Hope you understand, right? So this is what is the construction of M13mp1, right? So if you try to subscribe my channel, that's a Dr. K PREM-PRIMER for more updates. Thank you all. See you again with the one more video. Until then, bye-bye.